So our next learning unit is on glycogen metabolism and gluconeogenesis. So with these two processes, we can kind of think about them as being what we call a top-down and then a bottom-up approach to remedying low blood sugar. So for example, if you've got low blood sugar, you can tap into your glycogen stores in your liver and you can break that down in a top-down approach to generate glucose to remedy that problem. But again, gluconeogenesis is the process where we synthesize glucose from non-carbohydrate precursors, for example, like glucogenic amino acids. And we can use that as a mechanism for remedying low blood sugar. Both of these processes happen in the liver. And so we're going to study those two processes in some detail in this chapter. We'll also kind of delve then into how this process is regulated, and then a little bit on thinking about the biosynthesis of other carbohydrates. So what we're going to see at the beginning of most of these chapters that we have are kind of this 30,000-foot view, so we can remind ourselves where we are in the context of the entirety of several metabolic pathways. So what's central to this chapter is the understanding of glucose 6-phosphate as a branch point metabolite. So again, when we have branch point metabolites, they're defined as such because they've got lots of things that feed in or out of them. So to kind of talk about some of the things that we discussed on this slide, we talked about, again, glycogen being a reservoir and a storage of glucose in your liver, which helps to satisfy global needs, and in your skeletal muscle, which helps to satisfy local needs. So again, glycogen can be broken down via glycogen breakdown. We also call this glycogenolysis, so the lysis or the breaking apart of glycogen. And then the synthetic or the anabolic kind of route for this is remaking glycogen. For example, if we've got plentiful glucose and we can replenish those stores. So the bulk of this chapter is again dealing with this sort of large metabolic pathway substrate cycle, talking about either breaking down or synthesizing glycogen, but also sort of completing the metabolic or the pathway substrate cycle that we talked about. We learned about glycolysis in our last learning unit, and so now we're going to be focusing on gluconeogenesis, which is essentially the reversal of glycolysis. So this is kind of where we're going to situate ourselves for this chapter, highlighting kind of where we've been already. We've talked about the pentose phosphate pathway in our last learning unit, reminding you that the pentose phosphate pathway is important for synthesizing two main things: ribose 5-phosphate if we need nucleotides, and also NADPH for reductive biosynthesis, and also we talked about it for the purpose of regenerating glutathione. So highlighting here, glucose 6-phosphate is what you get as the breakdown product of glycogen. But if you want to export that, you actually need to remove the phosphate because only free glucose can be exported. So that's going to be an important point to highlight the differences in these metabolic pathways between different tissues, for example, with muscle and liver cells. We'll get to this a little bit later, but just to give you a preview, this chemistry, this biochemistry, this kind of dephosphorylation of glucose 6-phosphate by something called G6-Pase, glucose 6-phosphatase, only happens in the liver. The muscle cells do not actually have that enzymatic machinery to do that chemistry. So that's one of the reasons why skeletal muscle really only satisfies local or kind of selfish internal glucose needs. One of the other things that we'll kind of start thinking about too is we've talked about lots of different fates for pyruvate, right? We talked about homolactic fermentation, kind of what happens under anaerobic conditions. I'm going to highlight right now that this is a one-way path to convert pyruvate into acetyl-CoA, right? The enzyme is pyruvate dehydrogenase that does that chemistry. We're going to learn about it in our next learning unit in the citric acid cycle. But for us, for mammals, for our sort of metabolic processes, this is a one-way arrow. Fats cannot be made into glucose. But this is highlighting an important kind of feature of what happens nutritionally with something like the Atkins diet, okay? So with the Atkins diet, right, we can take, if you are not consuming carbohydrates, right, but you need to maintain 5 millimolar glucose, you need to make glucose from something else. We cannot make it from fats. So you must then make it from amino acids. So amino acids can be converted into pyruvate. These amino acids are specifically called glucogenic amino acids. They don't show an arrow here, but certain amino acids can be derived or made into acetyl-CoA. We call these ketogenic amino acids. So glucogenic amino acids can be converted into pyruvate. This is kind of a metabolically taxing process because, remember, amino acids have the element nitrogen, the atom nitrogen is part of it, so they need to be deaminated in order to only be carbon, hydrogen, and oxygen. So having to deal with all of that deamination chemistry can be taxing on some of your organs. And it's also a very costly process. However, lipids are worth a lot of money. So lipids, again, can be broken down to acetyl-CoA by that process that we learned of called beta oxidation. Converting lipids into acetyl-CoA and then allowing that acetyl-CoA to be used to make ATP is a great way that we can pay the cost for this process. So this, again, is the premise behind how the Atkins diet works. Now, there's lots of hormonal pieces to the Atkins diet that we'll touch on much later in the course here. But at its core, if you are on a low carbohydrate diet but need to make carbohydrates like glucose, you need to convert proteins like amino acids ultimately into glucose. That's a costly process, so lipids will pay that cost. And again, if those lipids come from your own internal stores, that results in weight loss. So just kind of a nice overview of some of the things that we've talked about already, kind of highlighting some of the things we're going to expand on not only in this chapter, but in future chapters. Okay, starting out here thinking about the structure of glycogen. So we've got kind of a nice cartoon here. Each of these spheres represents a glucose monomer. And one of the things that we'll highlight here is there's different colors that we see of different glucoses depending on where they are in this glycogen polymer. So kind of expanding on that structure down here, we've kind of highlighted the different colors on specific structures. So anytime you have an end of one of these glucose chains, that end can either be one of these blue spheres, which we call a non-reducing end, or one of the green spheres, which is a reducing end. So any end to the molecule represents a glucose molecule that's only connected to one other glucose molecule. So it's an end. It's not something that's in the middle of a chain. We're going to talk in a minute about these special ones that are actually connected to three different three different glucose molecules. But highlighting what we have here, a lot of these ends are blue spheres, which we call non-reducing ends. So again, this is an end. It only is connected to one other glucose residue. But if we look at the anomeric carbon, remember the anomeric carbon is the carbon that's connected in a glycosidic bond. That anomeric carbon is, again, part of a glycosidic bond here. So this anomeric carbon here is connected to a glycosidic bond, which makes this a non-reducing end. Let's differentiate that from this green one here. This is a special one. This is a reducing end. This guy is connected in a glycosidic bond, but it's a 1,4 glycosidic bond that's using its number 4 carbon. Its anomeric carbon is free. Okay, and what's important about this anomeric carbon being free, it's actually one step away from being a free carbonyl. Let's kind of highlight up here some chemistry that we've talked about. So if we look at, for example, one of these blue residues, and we think about this alpha 1,4 glycosidic bond, remember when we link sugars together in a glycosidic bond, we're making an acetal or a ketal linkage. So this carbon here is connected to 2 oxygen atoms. Okay? If we take and break that glycosidic bond, and we, for example, just have a free but cyclized sugar, we will have that anomeric carbon with a free hydroxyl group. So again, this sugar right here does not have a glycosidic bond on its C1 carbon atom. So it's a hemiacetal or a hemiketal. So remember, it's still a ring-closed sugar, but this sugar could actually ring open by breaking this anomeric bond right here. If we break this anomeric bond, we would have a free aldehyde or ketone. Remember, aldehydes and ketones are fairly reactive functional groups, so that's why nature cyclizes those sugars to protect that functional group. You learned in organic chemistry how you protect aldehydes or ketones is you make them into acetals or ketals, or at the very least, hemiacetals or hemiketals. That helps protect that reactive functional group. So what we'll see here is that there's always only going to be one reducing end. One reducing end. This is kind of where this glycogen polymer started. There's going to be lots of ways to have multiple non-reducing ends. And we'll talk a little bit later on about why having multiple non-reducing ends is important. But that handles the ends to our molecules, both non-reducing ends, and by glycogen having branching, we'll talk about that in a minute. That's how we have multiple non-reducing ends, and only one reducing end. Okay, so all of these glucose residues are connected by alpha-1,4 glycosidic linkages. So if you just kind of follow one strand here, all alpha-1,4 glycosidic linkages. We're going to have some special glucose residues here, the ones that are highlighted in purple. These ones not only have their alpha-1,4 glycosidic linkages, they're going to have an alpha-1,6 branch point. So these are what we refer to as branch point glucose residues. They're not non-reducing ends, they're not reducing ends, they are branch point glucose residues. So they actually have 3 glycosidic linkages. An alpha-1,4 off of its anomeric carbon, an alpha-1,4 to another glucose residue's anomeric carbon, and then an alpha-1,6 linkage to another glucose residue's anomeric carbon. But that creates a branch point from this this glucose residue. So one of the things that we'll note as we kind of move forward here, I used to have a homework problem that people would do where you actually had to count how many different kinds of residues that you'll see here. And what you'll find out is that about 10% of the total residues are these purple residues. These again represent branch points. So about 10% of glycogen exists as these branch points. And then the rest of these sort of nondescript are just sort of these yellow spheres, these yellow glucose residues, which just kind of represent the standard I'm in the middle of a glycogen polymer just linked with alpha-1,4 glycosidic linkages residues. So lots of those in a glycogen polymer. Most of glycogen's glucose exists as these ones that are just kind of in the middle, just alpha-1,4 linked glycosidic bonds with these glucose residues. All right, so that was a lot on structure. Let's move on and then talk about how we actually would break down this structure in order to get at the glucose that's there. So both glycogen breakdown and glycogen synthesis we're going to talk about is occurring in 3 steps. So I had you guys think about kind of specific names to help you remember these different steps. So we have a name for the step, the enzyme, and then kind of a brief description of what it does. So we're just going to kind of run through this quickly. The first step is mobilization. Mobilization is handled by the enzyme phosphorylase, which actually is the rate-determining step for glycogen breakdown. What phosphorylase does is it actually is going to cleave alpha-1,4 glycosidic linkages from non-reducing ends. That's not specifically written here, but that's a really important point to highlight, is this enzyme begins by finding a non-reducing end and chewing in and breaking alpha-1,4 glycosidic linkages, liberating free glucose 1-phosphate until it gets close to a branch point. We'll get to those details in just a minute. Once it gets close to a branch point, the next thing that has to happen is we have to start debranching our glycogen polymer. And that's because we can't have this enzyme continue to break alpha-1,4 glycosidic linkages when there's branches that are there. So the debranching enzyme is actually a multifunctional enzyme, a bifunctional enzyme, 2 activities. And the 2 things that it does is it's going to move branches and it's going to break alpha-1,6 linkages. So it's actually going to break the linkages between our branch point residues. The last thing that has to happen is that any glucose that was liberated as glucose 1-phosphate by phosphorylase needs to be made into glucose 6-phosphate. Remember, enzymes that move functional groups we call mutases. So phosphoglucomutase is going to convert G1P into G6P. And again, G6P is that important branch point metabolite that is going to be necessary to do any additional chemistries. So those are the 3 main steps, just kind of in an overview. We're going to talk about each one of them individually on a separate slide here. Okay, so glycogen phosphorylase. Here's the chemistry for what glycogen phosphorylase does. Glycogen, which is of a certain number of residues, is going to have phosphate essentially act as a nucleophile to break the glycosidic linkage, and you're going to have glycogen shortened by 1 residue and then glucose 1-phosphate. We call this chemistry phosphorylases because unlike using water as sort of the nucleophile, which would be hydrolysis, because phosphate is the nucleophile, we call it phosphorylasis. So what we can see here is here's this inorganic phosphate here. Okay, it's going to come in. We've got our glycogen polymer here. Here's the alpha-1,4 glycosidic linkage that's going to be broken. We actually need some vitamin B6 here. This vitamin B6 actually performs sort of a unique role. It doesn't serve as a source of phosphate, even though it is a phosphorylated compound. It's actually going to serve in a general acid, general base role, which is kind of, unique, not what you necessarily would have expected. But what it's going to do is it's going to serve as a general acid, and it's going to help increase the acidity of the inorganic phosphate that came in. That inorganic phosphate is going to protonate this glycosidic linkage, and that's going to facilitate, again, its departure its breakage using these internal electrons on the oxygen of the ring. We're going to generate this half-chair oxonium ion intermediate. Hopefully that name is familiar. We saw that same kind of chemistry. Remember when we had to have this half chair with lysozyme? We see something very similar here. It's going to help in that stabilization process. What we then have is PLP acting as a general base, is going to deprotonate this inorganic phosphate. That inorganic phosphate is now going to be a nucleophile attacking this electrophilic oxonium ion intermediate, and we're going to be generating glucose 1-phosphate here. So again, if this whole chemistry happened with water, we'd have an OH group here, and that's how we generate free glucose. But because this happens in this enzyme with phosphate, it's phosphorylasis, and we generate glucose 1-phosphate. So as we continue here through the course, we're going to see lots of times where we talk about mechanistic homologs or enzymatic homologs. What that really just means is, there's an enzyme that we studied already that has similar chemistry. So it's a great way to review topics that we've talked about already, but also kind of highlight some things that are unique and different and maybe seen in a different light here. All right, so that's glycogen phosphorylase. We're going to spend some more time studying glycogen phosphorylase because glycogen phosphorylase is the rate determining step of glycogen breakdown. All right, glycogen debranching enzyme. We're really not going to talk too much about any details of the enzyme active site. I do want you to know that it is a bifunctional enzyme that has a trans-glycosylase activity and then hydrolysis activity. So trans-glycosylase, so it's moving these limit branches, these glucose residues. So again, we talked about phosphorylase being able to chew in, right, from a non-reducing end. It's going to have to stop when it gets close to a branch point. I mean, imagine this is like a little Pac-Man that's coming in, and when it gets too close to kind of this congested area, it can't chew in any further. So what happens is this bifunctional enzyme is going to move what we call a limit branch. It's not moving this green residue here, which is the residue immediately adjacent to the branch point. This is the residue that has the alpha-1,6 glycosidic linkage. But it can move everything else, and what it does is just move it to the end of an existing branch, making that branch longer so that our little Pac-Man, our little phosphorylase enzyme can chew in from there. So that's the first activity, is this trans-glycosylase activity. The second activity is hydrolysis activity, and this is really unique. This is, again, the residue here. Even though it's a yellow residue, it's a residue that's kind of linked by an alpha-1,6 glycosidic bond. So again, it's next to the branch point glycogen or glucose residue. It's next to the one that has sort of 3 linkages. But this residue is actually going to have its glycosidic linkage broken by hydrolysis chemistry. Again, hydrolysis chemistry is going to generate free glucose. So this residue here is going to be liberated as free glucose. And why that's important is because every time we have a purple branch point, the yellow residue that was immediately adjacent to it is going to be liberated as free glucose. Remember, we said about 10% of the glucose that was found in glycogen are branch point residues. That means about 10% is liberated as free glucose. I want to make sure to highlight it's not the actual branch point residue, but it's the one that's connected to it via that alpha-1,6 glycosidic linkage. Last piece here is thinking about this concept that we've made a lot of G1P and we need to convert it into G6P because that is the branch point metabolite. So phosphoglucomutase is responsible for that. Okay, sort of some homologs that we've seen. We had phosphoglycerate mutase. That was one of those enzymes in glycolysis that interconverted 2-phosphoglycerate and 3-phosphoglycerate. Again, moving functional groups. It's basically going to move the phosphate from position 1 to position 6. So if we just kind of look at what happens in the enzyme active site, we actually have a serine residue that's phosphorylated. And it's not phosphorylated because it's regulating activity by kind of covalent modification. It's actually the phosphate donor. So in this case, what happens is we're going to have this phosphate get attached to the 6th position. We're going to generate this bis-phosphate compound, and then it's going to actually grab and take the phosphate off of the other position, leaving us with glucose 6-phosphate. Kind of mechanistically, the homolog that we've seen there, again, is phosphoglycerate mutase. And the reason that we need to do this is only G6P is that branch point metabolite we can do other things with. So some things that we'll spend some time talking about is if this is glycogen that you've broken down sort of for an internal use, that's going to actually result in a larger profit of ATP because you don't have to enter in that first use of ATP. Remember in glycolysis, the first step was hexokinase, adding ATP to glucose to make glucose 6-phosphate. So we can actually enter in the glycolytic pathway after that point. So we're going to be able to net 3 ATP after we've broken down G6P in glycolysis because we're not paying that first cost. However, if you're the liver and you actually have to transport this glucose out for external use, you actually have to dephosphorylate it. There's not transporters for anything except for free glucose, so you're going to use G6Pase, you're going to snip that off, and unfortunately you lose that benefit of having the higher energy phosphorylated compound. So it's kind of a cost, if you will, that gets paid if you're going to be exporting glucose, is you lose that initial sort of phosphorylated benefit. So moving on now to our synthesis pathway, we're going to consider synthesis happening in 3 steps. So like we thought about glycogen breakdown happening in 3 steps, we're going to think about 3 steps for synthesis. We kind of have to actually reverse one of the steps from glycogen breakdown or use one of those enzymes. We're going to use the phosphoglucomutase enzyme. We're actually going to need to reconvert G6P back into G1P. So once we've got G1P, now we're ready to continue with glycogen synthesis. So the first step we're going to call activation. If we're going to do anything where we're making new bonds, we often need to activate something, generally with a good leaving group, in order to allow that coupling chemistry to happen. So the enzyme UDP glucose pyrophosphorylase is going to take G1P and we're going to actually put a UDP group on it. UDP being a good leaving group, that's going to allow us to do some additional kind of coupling chemistry. The next chemistry that actually happens is the coupling chemistry. So we're going to call this step polymerization. The enzyme responsible for it is synthase. So synthase is the rate determining step for the glycogen synthesis pathway, and its job is to create alpha-1,4 glycosidic links. Now remember, if all we did was make alpha-1,4 glycosidic links, we just have one long straight polymer of glycogen that would have one reducing end and one non-reducing end. So what has to happen is the final step in glycogen synthesis is branching, because every time we add a branch, we're going to add a non-reducing end. So the glycogen branching enzyme does much of the reverse of what the debranching enzyme does, in that it creates alpha-1,4 or alpha-1,6 glycosidic linkages. So it's going to take sort of the end of one linear polymer and it's going to move and create one of these branch points by creating an alpha-1,6 glycosidic linkage. So not a lot of time we're going to spend on the enzymes in this synthesis pathway. We'll talk briefly about synthesis, but don't really worry too much about the details of these enzymes. So again, this UDP glucose pyrophosphorylase, we're going to need to generate that high energy glucose molecule, one that has a good leaving group. So again, we're going to use UTP. We need to use some energy for this. G1P, again, nucleophilically attacks this alpha phosphate of UTP, displacing pyrophosphate, and then we're going to add essentially this UMP group onto the G1P group. So one phosphate comes from the UTP, the other phosphate comes from G1P. That's how we end up with the 2 phosphates in UDP glucose. So a couple of things to highlight here that are helpful is we've got this inorganic pyrophosphatase. Remember, this is a theme that we see. Anytime that we generate this pyrophosphate, you've got enzymes in your body that are going to take and irreversibly kind of convert that pyrophosphate into free inorganic phosphate. It's a very exergonic process, so it drives this forward, and from Le Chatelier's principle, it's going to pull this whole reaction forward. So it kind of helps drive this whole activation process to completion. Again, just highlighting why this is important. We've got to activate this monomer for coupling by creating this good leaving group. So glycogen synthase. Okay, so again, we've got UDP glucose that's here. We're going to have this good leaving group leave, facilitated by departure with these lone pair of electrons on this ring oxygen. We generate this very electrophilic oxonium ion intermediate, and then that's very susceptible to nucleophilic attack by the C4 hydroxyl of a growing glycogen chain. Again, nucleophilic attack here is going to generate this alpha-1,4 glycosidic linkage. Now, it's important to highlight that this stereospecificity is what is imparted by this enzyme, because if we were to make a beta-1,4 glycosidic linkage, that's not useful for us. We need to have that alpha linkage in order for this to be fuel. So not too much more we're going to say about synthase right here, other than this is the rate controlling enzyme for glycogen synthesis. I will kind of talk briefly here about just these comments here, that if we imagine there being about 20,000 residues that we'd have in a glycogen polymer, if we were to consider that amount of glucose being in that tiny amount of space, the concentration of glucose would be about 400 millimolar. Remember that 5 millimolar is kind of physiological concentration for glucose in the blood. This would be such a huge concentration difference that the osmotic pressure would cause all cells to burst, as water would try to rush in to dilute this concentration. However, when you take all of those 20,000 glucose residues and you polymerize them into one molecule of glycogen, now that concentration of glycogen is 10 nanomolar. So the idea of your body sort of polymerizing and hooking things together is kind of disguising multiple particles into one or a few larger particles. And that really is important and helps when we think about kind of cellular osmolarity or pressure that can build up in cells because of concentration differences. The last thing to kind of comment is thinking about branching. So again, what's going to happen with branching is we're going to have synthase that has made kind of a lengthy branch that we have here. And what's going to happen with a branching enzyme is it's going to do kind of the reverse of what a debranching enzyme did, is it's going to take the end of a branch and it's going to actually reattach it to a C6 hydroxyl group here, making an alpha-1,6 glycosidic linkage and introducing a branch. So there's some rules that this enzyme has to follow. You actually have to leave at least 5 residues on an existing branch, so you can't make the branches too short. And you have to have some space. You have to have at least 4 residues from an existing branch point. So we can't have this be too crowded here, and we can't have our branches be too short. So all of these rules have to do with trying to optimize sort of 2 parameters here. We want to optimize the amount of glucose that we can store in a given space, but we also want to think about optimizing the speed with which we can get at that glucose when we need to break down that glycogen particle. So if we think about this storage space piece, right, to maximize storage space, we'd want to maximize the amount of glucose that we're going to have in a certain granule. That would mean that we're going to decrease the amount of branching, because if you have too much branching, it's going to get crowded really quickly and you can't get very large. Imagine this thinking about like a bush or something like that that's growing in your front yard. And if it's got lots of very dense branches, you can't grow very big before it starts crowding it out. And so you're not going to have as much branching. However, if we think about the other component here, that we want to be able to have the maximum mobilization speed when we need to get at the glucose that's in this glycogen particle, that would tell us that we want to have lots of branching because we want to maximize the number of non-reducing ends. So what we see here is we have these 2 things which are both important but sort of mutually exclusive in terms of how much branching you want to have. Really interesting that people have looked at kind of computational studies to try to determine what's the optimum amount of branching to both maximize storage as well as the number of non-reducing ends. And sort of the conclusions that they arrived at is, you know what, nature figured it out a long time ago and kind of got it right. I haven't asked you guys to think about what those numbers are, but there is specific sort of rules with how frequently glycogen is branched. All right, so moving on now to sort of think about regulation. And so we're going to think about the two enzymes that are really important for these two sort of parts of the glycogen substrate cycle. Again, glycogen phosphorylase being responsible as the rate controlling step for glycogen breakdown, and glycogen synthase being responsible as the rate controlling enzyme for glycogen synthesis. So as we've learned about with substrate cycles, right, you're not going to go downhill both ways unless one pathway kind of has the incorporation of energy. So we kind of see this in the synthase or in the synthesis sort of part of this pathway. And again, we need to use different enzymes in order to have these both be feasible. All right, so we're going to spend some time kind of reviewing an enzyme and a couple of enzymes that we've actually already studied. So again, I'm going to highlight here when we see things from previous chapters. So we've already learned about phosphorylase. And so again, phosphorylase is the enzyme responsible for breaking down glycogen, but we studied it in terms of it being either T state or R state. Remember, anytime you have an R state enzyme, that's going to be the one that's active because it has a conformation that is conducive to binding substrate and doing chemistry. So if we think about sort of this from a logic state, phosphorylase should be active when it's phosphorylated because this is sort of a need state pathway to break down glycogen. So that's going to be a phospho state. So this kind of just ran through kind of summarizing what we already knew about glycogen. And then this slide looked at the structure of glycogen phosphorylase to remind us of what we already knew about how we toggle between R state and T state. So for glycogen phosphorylase, remember, the conformation that it's in depends on whether this N terminus is buried and it's T state, or that N terminus is out and it's R state. So this enzyme is one that is subject to both covalent modification, that is the addition or removal of a phosphate, as well as allosteric regulation. So one of the things that we can see here is when we phosphorylate this serine 14, and this is again structural information that I do want you to know. So phosphorylation of serine 14 on the N terminus creates a negative charge, which prevents it from kind of wanting to be buried in the inside of a protein. But not only that, there's an arginine residue that's on the surface here. Remember, arginine being a positively charged amino acid residue creates a favorable electrostatic interaction that kind of locks it in the R state. So again, this phosphorylation chemistry is really what allows us to toggle between R state and T state. Reviewing another enzyme that we studied, again this time in chapter 13, we learned about pKa. Remember, this is the enzyme that toggles between an inactive tetramer. We've got our regulatory and our catalytic subunits, and it's regulated by cyclic AMP. So again, pKa is called protein kinase A, but it's also called CAPK, which means cyclic AMP dependent protein kinase. So when we have increased levels of cyclic AMP, that's going to bind to the regulatory subunits. The regulatory subunits will then dissociate from the catalytic subunit. Those free catalytic subunits can now do their phosphorylation chemistry. We looked at both the structures of the catalytic and regulatory subunits. So again, this is reviewing from chapter 13. So I'm going to go a little bit quickly through this, just kind of highlighting the important parts to know. So again, this is the catalytic subunit. Some important parts that we have here, we can see that we've got ATP bound in the substrate or the active site of the enzyme. This gamma phosphate is what's going to be transferred to the substrate. We can see here that we've got a threonine 197 that's in the enzyme active site. That's going to be toggled between a phosphorylated and dephosphorylated state. In order for this kinase to be active, threonine needs to be phosphorylated. And why that's important is because that helps to bind and orient the substrate. So we don't need to know too much about kind of the substrate binding recognition domain here. But I will highlight that this orange kind of representing kind of where the substrate binds, this is actually where the inhibitor segment on the regulatory domain will bind. So the regulatory domain, and we'll see this on the next slide, has an inhibitory domain, kind of a competitive inhibitor on its own protein structure that will bind to this enzyme active site. And if it's bound there, that means the catalytic subunit is tied up and it can't bind anything else to phosphorylate it. So here's again the regulatory subunit. The regulatory subunit has this purple domain here, and this purple domain is the inhibitory segment. So if this inhibitory segment is bound where the substrate would normally bind, then the catalytic subunit can't bind another substrate to do its chemistry. So that's how the regulatory subunit does its inhibiting. But if we've got cyclic AMP around, cyclic AMP sort of distracts this regulatory subunit. The regulatory subunit will bind to the cyclic AMP, it will dissociate from the catalytic subunit, and now the catalytic subunit doesn't have anything competing with anything else binding in its active site, and it can do its chemistry. So what we're going to spend some time studying here, and I will take a break at one point here and kind of skip over some slides that I've already posted a video on. We're going to talk about signal cascades. So one of the things that we'll talk about is a cascade that starts with protein kinase A. So we're going to say pKa phosphorylates phosphorylase kinase, phosphorylase kinase phosphorylates phosphorylase, and phosphorylase breaks down glycogen. So that's a tongue twister that when we first say it, it just sounds kind of crazy. But once you understand the process and this phosphorylation cascade, kind of all makes sense. So we're going to learn about the missing piece that we have here, the missing link between the chemistry of protein kinase A, or pKa, as a kinase, and then glycogen phosphorylase getting phosphorylated at that serine 14. So again, we've talked about it being phosphorylated at serine 14, but we really didn't talk about who did it. So phosphorylase kinase is who's responsible for doing that phosphorylating, and we're going to now understand how that happens. So we're going to do a lot to compare the differences between pKa and phosphorylase kinase. So just to highlight here, because we've got all of this up here, phosphorylase kinase is also a tetramer, just like pKa is, but it has 4 different subunits. It has an alpha and a beta regulatory subunit. So while in pKa these 2 regulatory subunits were identical, in phosphorylase kinase they're 2 different regulatory subunits. It has one catalytic subunit that we call gamma, and then it has another subunit here called delta, which is responsible for binding calcium. So here's what has to happen in order for the catalytic subunit in phosphorylase kinase to be active. We actually have to have phosphorylation of the regulatory subunits, so that's what we have shown here, is we're phosphorylating those regulatory subunits. pKa is actually responsible for doing that phosphorylating. And then we actually have to have calcium that binds to this delta subunit. So when both of these happen, when we phosphorylated the regulatory subunits, when we bind calcium to the delta subunit, we don't have a dissociation here, but we have a large conformational change that actually allows the gamma subunit to become active. So we'll talk about this on the next couple of slides, but how that happens is this gamma subunit actually has its own inhibitory domain. So imagine if you were to put your own hand in your mouth, if you will. That would inhibit you from biting or eating anything else. But if you underwent a conformational change and you pulled your own hand out of your mouth, then you'd be able to bind and eat anything else, which is essentially what this catalytic subunit is able to do when it's in its active state. I'll highlight down here that we're not going to talk too much about kind of the how PP1 becomes active, but I kind of want to just highlight here. We've talked a lot about phosphorylation cascades to kind of decorate things with phosphates, but it's just as important when we're in a fed state to have somebody who goes around and puts us in a dephospho state. So phosphatases do that. So the phosphatase that's really important in this whole cascade, in this whole process, is called PP1, which stands for phosphoprotein phosphatase 1. So PP1 is kind of one of these sort of universal phosphatases that's going to go and dephosphorylate a lot of things, just like pKa is going to phosphorylate a lot of things. So we're going to see PP1 in more than just this glycogen chapter. But again, we'll talk a little bit about kind of what happens with its activation, but not to the detail that I've got shown down here. So let's talk a little bit about phosphorylase kinase. And you have a homework question that kind of has you look at the similarities with what we see with the catalytic subunit of pKa. So again, what we have shown here on this slide is actually the catalytic subunit. So again, regulatory subunits, we're not going to look at the structures of them, but they are activated by phosphorylation. We've got kind of this gamma subunit that's going to be activated by calcium levels, and again, it's activated by binding those calcium to the gamma subunit. So let's talk a little bit about how this catalytic subunit is activated. It has a structure that's very similar to pKa. It's going to have ATP that's bound in the enzyme active site, ready to have the gamma phosphate transferred to its bound substrate. It has an inhibitory domain, just like we had with our pKa, but that was found on the regulatory subunit. This inhibitory domain is actually found within this subunit. So the only way to pull this inhibitory domain out is to undergo a conformational change that's going to again happen when those alpha and beta subunits are phosphorylated and when the gamma subunit binds calcium. So one thing to highlight here that's unique and different is when we have a glutamic acid residue here that's in place of serine or threonine that we see in pKa, this is what we call a phosphomimic. If you think about a glutamic acid residue, right? Glutamic acid residue is negatively charged, has a little bit of bulk that's in there. It kind of simulates what a serine or a threonine residue would look like if it was phosphorylated. So again, we've got pKa that's shown down here. So again, ATP that's bound in the enzyme active site, we see down here. We see this substrate binding site, which is where the regulatory subunit's inhibitory domain would bind down here. We see here this blue region is the autoinhibitor segment that we see within phosphorylase kinase gamma subunit here. And then here we had this threonine 197 that was phosphorylated in pKa. We've got a substitution to a glutamic acid, which means that this catalytic subunit is always on, right? Because if we put a glutamic acid in there, that mimics a threonine 197 that's phosphorylated. So some structural things here to think about with phosphorylase kinase. Not too much here to say about calmodulin. Again, this is the calcium binding domain. So this is kind of that fourth piece in that tetrameric phosphorylase kinase protein. Binds calcium. It actually is interesting. It undergoes large conformational changes when it binds calcium. So it really helps kind of change and shift the shape of phosphorylase kinase to make that gamma subunit active. All right, so this is PP1. Some things that we're going to talk about with PP1. I'm not going to go through the details of this. I'm going to kind of highlight a few things that are important, okay? One of the things that's interesting to think about is we would expect PP1 to be active under a fed state, right? Because if we're doing a lot, if we want to put ourselves in a dephospho system, that's sort of a fed state, so we're going to respond to a fed state signal. What's really strange here is we have a protein kinase that's stimulated by a fed state signal. Because again, kinases, decorating things with phosphates, you'd normally think, hey, that only happens when I'm in a need state. So again, this connection is a little bit different from lots of things that we've talked about, but the end product is the same, okay? Insulin is going to signal this kinase activity that's going to just put one phosphate on in a specific position, and it's going to make the most active form of PPI. So again, don't worry too much about this phosphorylation sort of state here. The end product is that insulin is going to activate PP1. What's important to highlight here too, because it's something we've seen before, pKa actually will phosphorylate this PP1 kind of complex as well, and it actually makes it inactive. So the net makes sense for our logic check. A fed state signal is going to activate our phosphatase. A need state sort of signal and pathway is going to actually inactivate our phosphatase. So the net product makes sense. How we get there can be a little bit confusing. So I'm not going to say too much. I'm going to flip through the next slides here because I've actually already put a video together that kind of highlights the differences between these phosphorylation and dephosphorylation systems with both high and low blood sugar. And that's what this is. And then thinking about how high and low blood sugar is going to be responded to differently by muscle versus liver cells. So again, I'm going to skip through these slides. Make sure to go back and watch that separate video to kind of get the details of this. All right, so the last thing to sort of talk about in this chapter is gluconeogenesis. So we've spent a lot of time there talking about the top-down approach to remedying low blood sugar, but that's only if you've got glycogen reserves. What if your glycogen reserves are gone? So again, if your blood sugar is low and you've basically used up all of your glycogen reserves, the only option is to synthesize glucose from scratch. Okay, to highlight here, we cannot make glucose from fats. But the net of what happens here is we're going to run glycolysis in reverse. So to highlight here, remembering glycolysis is 10 steps. So anything that was a large negative delta G in glycolysis, we can't go downhill both ways. So we have to choose a different path. And again, we have to have one of those paths include the incorporation of energy. So if we think about, remember when we talked about the regulation of glycolysis, we asked what steps were far equilibrium. It was steps 1, hexokinase, step 3, phosphofructokinase, which is again the rate determining step in glycolysis, and then pyruvate kinase, the last step in the pathway. So we need to find alternative paths for just those 3 steps. Everything else we can use the same enzymes. So truthfully, we don't even really need to talk too much about these 2 systems because we're just going to use a phosphatase enzyme. So it's just a different pathway. This first step in reversing glycolysis has so many different pieces to it that we're actually going to spend the bulk of our time here sort of talking about that. So kind of the chemistry to convert pyruvate into phosphenylpyruvate, it involves like multiple enzymes, multiple locations. And so I'm going to kind of give you the overview here, and then we're going to get into some details. So converting pyruvate to oxaloacetate actually involves the addition of carbon dioxide. It's going to cost us some energy. And importantly, this only occurs in the mitochondrion. Everything else here, remember glycolysis occurs in the cytosol. So everything else that we can reverse here, fine to do in the cytosol. We have to dip into the mitochondria for a little bit here because this enzyme only lives in the mitochondria. Okay, so that's problematic. I want to kind of highlight here with what we've got shown here why we actually need to make oxaloacetate. Oxaloacetate is something we're going to talk about more in our next learning unit on the citric acid cycle, but this is again another one of these branch point intermediates. Lots of things might feed in or out of oxaloacetate. But if we look at pyruvate, essentially what happens with pyruvate is we need to get to the point where this methyl group becomes nucleophilic. We want to have it be a nucleophile so that we can actually push those electrons up through this oxygen and then attach a phosphate there. That's what phosphenylpyruvate looks like. A common theme that we're going to see in kind of creating biological nucleophiles are to add carbon dioxide to create what we call a beta keto acid, and then decarboxylation of that beta keto acid makes this carbon atom have a negative charge, which we either can have that directly be a nucleophile, or again pushing those electrons up through this oxygen atom, allow that oxygen atom to be nucleophilic, reacting with something like GTP to add that terminal phosphate here. So if that didn't make sense, we're going to see the details of that on the next couple of slides here. So again, 2 steps to kind of convert pyruvate to phosphenylpyruvate. The first step uses an enzyme called pyruvate carboxylase. So what's going to happen is we're going to have bicarbonate that we're going to pull from our bloodstream. We actually need to use another one of our B vitamins here. We're going to use vitamin B7, which is biotin. That makes what we call transferable CO2. So what we're going to do is we're going to actually put CO2 on this terminal methyl group. That generates oxaloacetate. Oxaloacetate, again, being our branch or our beta keto acid. So don't need to really worry about the structures here, but what vitamin B7 does is inside the enzyme active site of pyruvate carboxylase, this biotin is going to be attached to a critical lysine residue, and it's going to take that bicarbonate and attach it as a CO2 group here. And that just allows that to be sort of a hot potato transferable CO2. Remember, and we'll get to the details of this, this only occurs in the mitochondrion. So we had to shuttle pyruvate into the mitochondria, and then we're able to do this pyruvate carboxylase chemistry. The next step is handled by an enzyme called PepsiK. So PepsiK is going to take oxaloacetate that easily decarboxylates because it's a beta keto acid. And again, Taking these electrons, pushing them up through this oxygen will allow us to nucleophilically attack the electrophilic gamma phosphate of GTP, essentially transferring this terminal phosphate to this oxygen atom and generating PEP. So a common theme that we'll see when we do chemistries where we need to generate a biological nucleophile is we're going to see the addition of CO2 and then its removal to generate our nucleophile. Okay, so one other thing that we got a little bit deep on was talking about how we kind of handle all of this shuttling. So I'm going to have 2 slides here to kind of summarize that. But one of the things to kind of talk about is, and it's not shown on this picture here, we do have a transporter to get pyruvate in and out of the cytosol and the mitochondria, so between those 2 kind of different locations. So pyruvate can easily go back and forth. So can something called malate and aspartate and phosphenylpyruvate. So we've got transporters for all 4 of these guys. What we don't have a transporter for is oxaloacetate. So again, the enzymes to synthesize oxaloacetate are mitochondrial. So both pyruvate carboxylase as well as PepsiK live in the mitochondria. But the enzymes to make phosphenylpyruvate, again, are exist in both. I may have misspoken there. Oxaloacetate, the enzymes, again, for its synthesis are only mitochondrial. So pyruvate carboxylase exists only in the mitochondria. PepsiK exists both in the mitochondria and the cytosol. Because glycolysis is a cytosolic process and we're essentially reversing glycolysis to do gluconeogenesis, we kind of need to dip into the mitochondrion for a little bit for pyruvate carboxylase, but then we need to finish the process out in the cytosol. So this is important in terms of looking at the specific enzymes, and we're going to talk about that in a second, but then I'm going to highlight another figure that we put together in class that I think kind of summarizes things a little bit better. All right, so this is a common theme we're going to see. If you don't have a transporter for a specific metabolite, what you can do is disguise it as something that you do have a transporter for. So oxaloacetate can be disguised as 2 other molecules. You can have oxaloacetate that you convert to malate by doing some reduction chemistry. Okay, so again, reducing this carbonyl here to an alcohol converts oxaloacetate to malate, which then you can come out into the cytosol. Now you have to make sure that you've got a cytosolic version of malate dehydrogenase to reverse that chemistry and get back to the guy that you really wanted to be outside. Alternatively, oxaloacetate can be converted into amino acid by doing some transaminase chemistry. So aspartate aminotransferase, so AAT is what this enzyme is called, can actually take oxaloacetate and you convert this carbonyl essentially into this carbon bonded to nitrogen. That makes the amino acids aspartate. There is a transporter for aspartate. You can get that out into the cytosol, and as long as you have a cytosolic AAT enzyme, you can reconvert aspartate into oxaloacetate. So now we've got oxaloacetate out in the cytosol. It's ready to go through and do gluconeogenesis. One of the things to kind of highlight here is you also have the ability, remember that PepsiK, that's the enzyme that converts oxaloacetate into phosphenylpyruvate, that exists in the mitochondria. So you can take oxaloacetate, convert it into phosphenylpyruvate, and then shuttle that out to continue gluconeogenesis. So I'm going to talk about that same information just on a different sort of picture here. So I want to highlight that pyruvate can kind of come from 2 different places. You can have pyruvate that maybe came from glycolysis at some point. And again, glycolysis may be of some other non-glucose carbohydrate. Remember, lots of other carbohydrates can feed into glycolytic sort of intermediates. So if you've got pyruvate that you made in the cytosol, you'd have to get transported into the mitochondrion in order to start this process. Or maybe you have some glucogenic amino acids that were broken down and kind of made maybe into pyruvate within the mitochondria. So maybe you already actually have pyruvate in the mitochondrion. Regardless, this is the key step here. Pyruvate carboxylase only lives in the mitochondria. So to convert pyruvate into oxaloacetate, you have to be in the mitochondria. Now, Once you've made oxaloacetate in the mitochondrion, you have 3 options. And again, those were highlighted on the previous slide, but I'm going to kind of run through them here. And if you need to, rewind the video and go back and see those same pathways sort of mapped onto the physical transporters there. One sort of simple option is just say, you know what, I'm going to finish this chemistry in the mitochondrion. PepsiK lives in the mitochondrion, so I'm going to use that to convert oxaloacetate into phosphenylpyruvate. And now I've got a transporter to get phosphenylpyruvate from the mitochondrion out to the cytosol. And once I've got phosphenylpyruvate in the cytosol, then reversing glycolysis is pretty easy. Gluconeogenesis is going to make the glucose that I need. However, you can actually choose not to finish that conversion in the mitochondrion. You can disguise oxaloacetate into something else. And again, this is called the malate aspartate shuttle because basically oxaloacetate can be converted into either malate or aspartate. So converting oxaloacetate into malate requires a mitochondrial version of malate dehydrogenase. Then we can utilize the malate transporter, get malate out into the cytosol, and then as we need to be able to have a cytosolic version of MDH to then reverse that chemistry and make oxaloacetate out in the cytosol. Another sort of route you could have is oxaloacetate could be converted into the amino acid aspartate in the mitochondrion. Again, that's going to be this mitochondrial version of AAT. We can then use the aspartate transporter to get out into the cytosol. Once we're out in the cytosol, then the cytosolic version of AAT can reverse that chemistry, regenerate oxaloacetate in the cytosol. Now remembering that PepsiK lives both in the cytosol and the mitochondrion, if we have the cytosolic version of PepsiK, it can finish this conversion of oxaloacetate into phosphenylpyruvate, and now again we're ready to reverse glycolysis, complete gluconeogenesis, and make the glucose that your body needs. So our last little piece here is to talk about sort of how we regulate gluconeogenesis. And in talking about regulating gluconeogenesis, we're sort of indirectly talking about regulating glycolysis as well. Because remember, going between glucose and pyruvate and back to glucose really represents kind of one large metabolic substrate cycle, kind of cycling through an entire pathway. So glycolysis, and then gluconeogenesis. So we already knew that PFK is how we regulate glycolysis. So not surprisingly, its companion enzyme in the gluconeogenesis pathway is how we regulate gluconeogenesis. So how we regulate this entire process really has to do with how we regulate both PFK and FBPase. And I'm going to find and flip back to the slide that we had about PFK regulation here. So remember when we had this slide, we talked about regulating PFK. So we just talked about regulating PFK by T state and R state, and we talked about kind of binding small effector molecules, and we talked about binding ADP and so forth, and kind of swapping in and out a glutamic acid residue in an arginine. So that's important, and we kind of regulate via binding allosteric molecules. But one of the things that we didn't necessarily talk about is how we can sort of superimpose and map on phosphorylation aspects to this. We don't have a direct phosphorylation site on either of these enzymes, so it's not like a kinase or a phosphatase directly kind of controls what's going on with this enzyme. But what we do have is either kinase or phosphatase activity controlling the synthesis of F2,6P, which again is not a glycolytic intermediate, but it is a small molecule that is going to regulate the activity of the two enzymes that are responsible for controlling flux in glycolysis and gluconeogenesis. Okay, so F2,6P, we actually talked about this when we did our glycolysis chapter, and all that I asked you to think about was the fact that it is a fed state signal. Okay, it is something that's actually going to be activating glycolysis and inhibiting gluconeogenesis. And I know it's a little bit weird to think about, wait a minute, glycolysis is a fed state pathway? Do not confuse an energy need with Glucose breakdown. So we're not going through glycolysis because we're in need of energy. We're actually going through glycolysis because we need to do something with that excess glucose that we have. We need to kind of bring our high blood sugar levels down. So you need to metabolize it, which means you need to convert it into pyruvate and then ultimately into acetyl-CoA and made into fat stores. So a way to help remember this too is, I think it's obvious that gluconeogenesis is a need state pathway. So if gluconeogenesis is a need state pathway, by default, glycolysis must be fed state. So kind of, we had a handout that we had in class. We talked about kind of a confusing set of need states. Again, glycolysis really at its core is a fed state pathway in terms of how it's regulated. Okay, so F26P is going to be activating glycolysis and inhibiting gluconeogenesis. So let's see if we talk about how this allosteric regulator, this effector molecule, is made. That's consistent with what we'd expect. Okay, so let's see if we can put this together here with what we can think about in terms of phospho versus diphospho state. So this enzyme, this bifunctional enzyme, basically has kinase activity and it has phosphatase activity. So again, if F26P is a fed state signal, we'd expect it to be made in a diphospho state. And again, this is one of those, it's a little bit weird, right? We've got kinase activity under a diphospho system. So again, the kinase part of this enzyme is active under a diphospho state, but the end product makes sense. When we're actually doing phosphorylating here, we're actually going to be phosphorylating the 2 position on F6P, and we're generating F26P. But that makes sense. Again, diphospho system, we're going to be activating glycolysis because that's responding to that fed state signal of high glucose. Again, let's contrast that to when we've got a phospho system. When we've got a phospho system, this enzyme is going to have its phosphatase activity be active. When its phosphatase activity is active, we're going to be making much more F6P. And again, that's going to be decreasing the concentration of F26P, decreasing that fed state signal. We're actually going to be increasing gluconeogenesis. So one of the really interesting things here to kind of piece together is some of the players that we've actually already seen before. pKa and pp1 are the ones that are really phosphorylating or dephosphorylating this bifunctional enzyme. So, oh my gosh, the layers that we have here just kind of can get a little bit complicated. But if you remember that F26P is a fed state signal, and then the bifunctional enzyme that's kind of responsible for either making through kinase activity or decreasing the concentration of F26P by phosphatase activity, we can sort of kind of put this all together with a logic check. And so that's kind of what this is. You have this question sort of asked as both a high and a blood sugar on your high and low blood sugar on your homework, but just kind of running through here. Again, if we've got low blood sugar, your pancreas is going to be secreting glucagon. Glucagon, being a need state signal, is going to activate adenylate cyclase. That's going to increase cyclic AMP concentration. Increased cyclic AMP concentration is going to actually increase pKa activity, so enzyme phosphorylation through pKa. Remembering, if we're doing this phosphorylating, we're actually going to be activating the phosphatase activity in that bifunctional enzyme. Activating the phosphatase activity is actually going to be decreasing the amount of F26P that we have. So now if we're decreasing this fed state signal, that normally would have been a signal that activated glycolysis, so we're going to be inhibiting glycolysis, activating gluconeogenesis, and again, the net outcome is increasing the concentration of glucose we have as it's synthesized by the liver via gluconeogenesis. Wow, that last piece can be, really, really complicated. So walk through it a few times, kind of make sure that you sort of see the logic check. There's 3 things that are kind of dense and meaty in this chapter. They're all related to thinking about high versus low blood sugar. So I kind of want to just highlight again what they are here for you. High versus low blood sugar, thinking about how we operate under a phospho or a dephospho system with these cascades. High versus low blood sugar, and thinking about how these two different tissues respond. And then lastly here, high versus low blood sugar, and thinking about how we regulate gluconeogenesis versus glycolysis. Okay, so 3 sort of meaty, dicey, really involved things there. So make sure, look at the homework. It's posted online so that you can kind of review those kind of intense portions. The last little piece here to kind of highlight, and it's more just almost like a little side note here, but it kind of brings in some of the things that we've talked about already. Remember when we talked about glycogen synthesis, we talked about the need to have an activated monomer in order to do that coupling reaction. That's kind of a common theme that we're going to see. If we're going to be making glycosidic bonds, we basically have to activate the sugar to do so. Generally, how we activate that is we add nucleotide diphosphates to make a good leaving group. So again, we're going to have some acceptor sugar. It's going to come in. It's going to be able to nucleophilically attack this anomeric carbon, probably after this has already left and departed and created that very electrophilic kind of oxonium ion intermediate. But one of the things that's just interesting to kind of highlight is the identity of that nucleotide sort of diphosphate can give some information as to what that glucose monomer is supposed to be doing. In mammalian glycogen, we talked about UDP glucose being the monomer. If you're a plant, you actually either have ADP or CDP that's going to be present. And the identity of that nucleotide really dictates whether you're making alpha-1,4 glycosidic linkages and you're going to be making starch, or beta-1,4 glycosidic linkages and you're going to be making cellulose. And there's lots of different NDP sugars that are going to be used to either do N-linked or O-linked kind of chemistry. And again, when we talk about using and decorating proteins via post-translational modifications, we need to have an activated sugar in order to do that. One of the things we didn't really talk about too much in class, but just kind of an interesting little side story, is your body's ability to make lactose, which is milk sugar. So you basically make lactose by mixing galactose and glucose together. You activate the galactose residue by making it, again, be the one with the good leaving group. And then generating that beta-1,4 glycosidic linkage is how we're going to make glucose. What's kind of interesting is that you normally have these building blocks in your body, but you don't have the enzyme responsible for doing so until you have a hormonal signal that allows you to make that enzyme that's going to couple these guys together. What's really interesting is the ability to have that that subunit be made to kind of do this synthesis is not gender specific. If given the right hormone, either male or female mammals can be induced to lactate. So yes, we can make men lactate. Just a little bit here, we didn't actually go through this in class, but thinking about, we talked about it, but thinking about some mechanism here, right? If we're talking about breaking the glycosidic linkage that we're going to have in a sugar, remember making a cyclized sugar is how we make a hemiacetal or hemiketal. Making a glycosidic linkage is making either an acetal or a ketal. So if we want to talk about breaking that glycosidic linkage, we need to protonate our leaving group, make it be a good leaving group. Internal nucleophile is going to push that out, generating, this would be the equivalent of our oxonium ion or a half chair intermediate. Nucleophile is going to come in. Again, if our nucleophile is water, we've done hydrolysis of our glycosidic bond. If our nucleophile is phosphate, that's what we see with phosphorylases. That's what we see in our phosphorylase chemistry. Thinking about chemistry that happens with synthase, right? Again, if we want to take a hemiacetal or a hemiketal, we've got an end, a reducing end that maybe needs to be linked to another glucose monomer so that we can kind of grow our polymer chain. We're going to see a good leaving group leave here. And again, we're highlighting here that if we had a nucleotide diphosphate sugar, this would be a good leaving group, not water. we would generate this very electrophilic oxonium ion intermediate. Again, if this is kind of our cyclized sugar, we've got our half-chair oxonium ion intermediate. Depending on what the nucleophile is that's coming in, if it's another sugar, we're going to be able to create a glycosidic bond. If we're creating this from the C4 hydroxyl position, that's going to generate our alpha-1,4 linkages. If it's from the C6 hydroxyl, that's how we're going to generate our alpha-1,6 branch points. So again, mapped on here in red is kind of the typical reaction mechanism that we saw in Biochem 1. And the colored regions kind of highlight how it's a little bit different when we talk about using the functional groups that we see in this chapter. So again, cleavage of glycosidic bonds, that's what happens with phosphorylase. And then formation of glycosidic bonds, that's what happens with synthase. And that is the end of our glycogen metabolism chapter.